Hi everyone, I'm Dr. Anisha, your microbiology faculty, and today I will be discussing about an important virus, that is influenza virus. And under influenza virus, I will be covering up antigenic drift, antigenic shift, along with genetic reassortment and different pandemics that this virus has been associated with. Now, firstly, when we talk about influenza virus, we must know that it is a myxovirus. Influenza is a myxovirus, right? And myxoviruses can be divided into two families, that is 𝗤𝗏𝗡𝗅𝗏𝗡𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 𝗘𝗅𝗕𝗃𝗏𝗏 as well as 𝗕𝗵𝗵𝗀𝗏. Now, an important differentiation feature between these two families is that it is the 𝗘𝗕𝗵𝗄𝗏 family which contains segmented RNA. Segmented RNA. Right? So now let us come to the different important features associated with influenza virus. Firstly, it belongs to which family? This virus belongs to 𝗘𝗕𝗵𝗄𝗏 family. And the morphology, for understanding the morphology, we first must be talking about the size, then the symmetry, then different structural proteins, the segments which are present inside, etc. So let's first talk about the size. It is around 80 to 120 nanometer. And this virus has helical symmetry. Helical symmetry. And let's consider it from the name itself that influenza, it's an enveloped virus. It's an enveloped virus, whereas it is associated with different flus. The most important and the most famous being swine flus, avian flus, etc. So influenza, it is very well associated with flus and it is an enveloped virus. And it has different structural as well as non-structural proteins. And non-structural proteins. And most important feature being the segments of the RNA. The RNA is segmented. And the multiplication of RNA takes place inside the nucleus, which is not the case with other different RNA viruses. So RNA replication takes place in the nucleus itself. So about the different structures, we'll be seeing them in the viral proteins and we will be discussing about the different morphological features from the diagram itself. But this is the way how influenza virus looks like. It has segmented RNA, different segments. These are the segments of RNA, right? And then we have the envelope. This is the envelope. And the other structural proteins we will be covering up with the same picture in the later section. Now, influenza virus, it is divided into 4 genera, important ones, A, B, C, and D. And how do you know and how do we describe the number of segments that influenza virus? So we have already discussed that influenza has 4 genera, A, B, C, and D. So how many segments are present in A, B, C, and D? How do we differentiate? So A and B influenza and B has 8 segments, B is the later letter, right? So B is, its structure is similar to 8. So A and B influenza genera have 8 segments, whereas C and D, so C can be write 7 for C and 7 instead of S, right? So C and D have 7 segments. Right? Now moving towards the viral proteins. Now the virulence of influenza virus is majorly associated with two important viral proteins, that is HA and NA, and we will be talking about different functions, smaller functions of the different structural as well as non-structural proteins. So the viral proteins can be divided into structural and non-structural proteins. So firstly, to understand the structural proteins, let us consider this mnemonic, 𝗏𝗏𝗏. So Hannah Montana was a pop culture show which was very famous in our childhood days. So Hannah Montana was a pop singer, right? So she was a pop singer and she loved going to the pubs. So how do we use this in our structural proteins? So M, considering M, it stands for M1 and M2 proteins, right? Then HA and NA are the next structural proteins we will be discussing about. So HA and NA. These are two different structural proteins. Then we'll take P and B from pub. So this comes down to PB1 and PB2. Then we have another, this we'll have to remember, we'll have to add PA. And the last one, structural protein, is going to be NP. So what are these structures defined as, and what are the important roles of these structures? Firstly, M1 and M2 are the matrix proteins. These will be responsible for viral entry and the destruction of the host surfaces, right? And they'll help the virus entering inside the host cells and causing the destructive effects. So hemagglutinin and neuraminidase. These are hemagglutinin and neuraminidase. These are glycoproteins and the most important structural proteins which have been associated with pandemics, epidemics, and any forms of outbreaks. Then come PB1, PB2, and PA. Now these are responsible for RNA transcription and replication. And NP, it is the main capsid protein, major capsid protein. These are all the structural proteins and their important functions. Now coming to the non-structural proteins, we have two non-structural proteins, NS1 and NS2, right? So these you all need to remember what are the structural and non-structural proteins and what are their major functions. Now moving towards the most important structural proteins, that are HA and NA glycoproteins. So let's first see the structure. First look at the image and see what these proteins look like. So considering this triangular structure, this triangular structure, this is the hemagglutinin. HA hemagglutinin. Now, since it is triangular in structure, so it somehow helps us identify that this is going to somehow allow the viral entry. Triangular sharp structure. So this allows viral entry. And it binds to sialic acid receptors. Sialic acid receptors on epithelial cells. And which epithelial cells? Specifically, respiratory epithelial cells. Right? So first thing is this, hemagglutinin is one structural protein or one glycoprotein which is helping the virus enter inside the host cell. Right? And then the other important structural protein, which is this mushroom-shaped structure. This mushroom-shaped structure, which is nothing but the neuraminidase. Neuraminidase, and it has a sialidase enzyme activity. Sialidase enzyme activity. So what is it doing? It is naturally destroying the sialic acid receptors. Destroying the sialic acid receptors. And further facilitating the virus entry and the destruction of different receptors and structures lying around. Right? And when you're talking about the matrix proteins, we have M1 and M2. So this M1, these round structures present beneath the envelope. This is the, say for example, this double line blue structure, this is the envelope. And the layer beneath the envelope are these small globular structural rings. This is nothing but M1, matrix protein 1. So what is its function? It is just forming a shell beneath the envelope. And then we have another structural protein, M2. This M2, this anchor-like structure, M2, which is forming a channel. Does it look like a channel which is formed inside the lipid envelope layer? Right? So this is forming the ion channel for movement of molecules, for molecular movement. Across the structure, across the virus, right? And the transit of different molecules from the host to and fro to the virus protein or the viral structure, right? So these are the important structural proteins, and the one which we have already discussed, RNA segments. These are different RNA segments. And to consider it, whether it is influenza A or B, it should have 8 segments, and C and D are of course going to have 7 segments. Now let us discuss a little bit about antigenic subtypes or antigenic genera, which we have already discussed, that are A, B, C and D. Let us rule out the non-important ones first. So D, these are associated with animals and birds and do not cause human infection. No human infections. So no further discussion is needed for this. Then C, these are associated with milder infections, and are not associated with any public health concerns. No public health concerns. Right? And then we have genera B, which are not divided into subtypes, but they have been transformed into different lineages. They are further taken into lineages, and these can be Victoria lineage, B, Victoria lineage, or B, Yamagata lineage. And A, of course, this is the most important subtype or genera. And it is further divided into different subtypes based on which structural proteins. These are NA and HA. So whenever we talk about influenza virus, these are the most important ones that we need to discuss, right? So see how HA and NA subtypes are further divided and how many subtypes have till now been discovered and are associated with human infections. So Hannah, she's 18 years old, but at that time she never used to look 18 years old, but she looked 11 years old. So this comes down to the number of HA and NA subtypes. So always remember Hannah as an important character for influenza virus. So 18 HA subtypes, that is H1 to H18, whereas NA subtypes are N1 to N11. So these are the total number of subtypes, and most of these subtypes are associated not only with human beings but also with animals and birds. Animals, birds, and human beings. Now comes the question that what are the different subtypes, or rather important question, what are the different subtypes that have been isolated for human beings or are associated with human beings? So these human subtypes are, firstly let us talk about H and then we'll discuss about N. So remember, for H we'll take up all the odd numbers first. So the ones discovered from humans are H1. And only H2, it is the odd one out because it's an even number. So the next odd number, it is H3, H5, H7 and H9, right? And what are the two N subtypes which have been isolated for humans? It is N1 and N2. Easy to remember. So remember, all the odd numbers for H subtype and N1 and N2 for N subtypes. So it is very important for influenza as an infection or as a condition which you should know that these take place mostly in winters. And seasonality, it is an important point when we talk about pandemics, right? Now let us consider another important topic for influenza virus, that is antigenic variation. Now antigenic variation, it is a unique property which is exhibited by influenza virus because of certain changes or alterations in amino acid sequences or any antigenic changes which take place in the structural proteins HA and NA peclomers. So it is a property which is exhibited by the influenza virus because of changes in HA and NA peclomers. And what kind of changes are these? These are antigenic changes. So these antigenic variations can be classified into two types. One is antigenic drift as well as antigenic shift. So now to understand the mechanism of these different variations, let us first consider two important stories and let us help them take up into the mechanism. Let us take these stories into mechanism to better understand them. So let's first talk about antigenic drift, and we are considering here a couple who has minor fights taking into consideration silly points or topics. So because of these frequent silly fights, nothing major happens, but there is small disagreement or indifferent behavior towards each other. And these fights are very frequent in nature, so they occur every 2 to 3 years. But nothing major or drastic is happening here. So let us look at the mechanism. So firstly, the mechanism goes like there are point mutations which occur in HA and NA peplomers. So because of these point mutations, there are alterations in amino acid sequences of the HA and NA proteins. So there are only certain alterations which are occurring, nothing major is happening. So because of these alterations, these escape partial recognition from the host cells or host immune system. Now they have escaped their recognition. So now they have the ability to multiply and will not be killed up by the host cells. So there are only certain amino acid alterations which are occurring, right? So no new virus is being formed, no new strain is being formed. It is just temporary variation and it is seasonal. It can occur, it is frequent in nature, it can occur every 2 to 3 years. And these are not associated with any major drastic infectious series or any major drastic pandemics or something like that. So what is occurring here? Suppose this is virus A and it is only some change that has occurred inside the virus, majorly in the structural proteins, which has led to only small variation in the viral structure, and there forms virus P, right? So no new virus is being formed. It is small alteration which is responsible for the infection. So now comes the question that which influenza genera show antigenic drift. So it is influenza A as well as B which are associated with antigenic drifts. So these result in small outbreaks as well as minor epidemics. And from the story itself, we could derive the frequency in occurrence. It is every 2 to 3 years and they are frequent in nature. Please consider this couple theory or couple story as the mechanism discussion. So when we discussed about the minor fights because of silly points, we could make out that it is the point mutation which is occurring and they are frequenting every 2 to 3 years. So this is the occurrence and no nothing major is happening, so it is responsible for minor outbreaks. So now let us discuss about antigenic shift. So for antigenic shift, let us also consider another couple, or rather fierce couple. So this couple now somehow had a major fight and drastic changes took place. They stopped talking, they moved places, and they started living separately. And they started having a different life. So now, while being separated, they started going for some temporary flings, and they kept analyzing their flings. And after analysis, they figured out that they have become a totally new person. They have a very new, different life. And now, nothing from past affects them. I think this happens in most of the cases, that somehow people grow apart, right? So let us take this fun story as a part of our mechanism understanding. So what is happening here? So there are abrupt, sudden, major, drastic, and discontinuous variations which are occurring in the amino acid sequences, or the sequences in the structural proteins. And what are the structural proteins we have discussed? It is always going to be HA and NA. And what is this? This is leading to formation of a totally different virus. So, and what is the real mechanism behind that is? It is the genetic reassortment. And because of genetic reassortment, so there are two viruses. There can be one, there can be two, there can be three, there can be more viruses. Now these two viruses, from this image, let us understand that these two viruses, they are affecting a single host, right? They are affecting only one host, and because of the drastic or major changes which have occurred, this leads to formation of a totally different virus now. This virus C has been formed, and why is that occurring? Because of genetic reassortment. And what is genetic reassortment? There is complete swapping of the segments, RNA segments, between two or more viruses. So it is not just one or two mutations which are occurring in the amino acid sequence. The whole segment, it becomes interchanged, and there is discontinuous variation. There is not a pattern variation which is occurring between two different virus strains affecting the host itself. It is discontinuous variation, very haywire things, very unconventional things that occur, leading to formation of a totally new virus which is not at all associated with virus A, virus B, or the previous strains which were affecting the host cell. So this new virus, it is totally new virus, and it has no association with its parent viruses. In this picture, we have virus A and virus B. There can be more viruses as well. So can you see the difference? We have different papillomas, different colors, we have different RNA segments altogether. So a new virus has been formed. So this is how we can use our story. So the couple was living a new life, and nothing from the past has now affected them, right? So this new virus is completely different from its parent viruses, and the antibodies which were previously effective against virus A and B have now become ineffective to the new virus, virus C. So nothing from the past, that is no parent cell similarity, and no previous antibodies are now effective against virus C. A totally new virus has been formed. Virus because of drastic variations and discontinuous variations in the amino acid sequences, right? So now the most important question, that which virus genera is associated with this major drastic change? It is only and only Influenza A. Influenza A is only associated with antigenic shift, which results in, now we have already discussed that major change is occurring, major formation, major variation is occurring, which is leading to formation of a totally new virus. So whenever a new virus comes in our society, so it leads to a state of pandemic, a major situation that takes place, right? That is the case which occurred in coronavirus, although it is not associated with 𝗲𝗵𝗄𝗏 family, but that was a pandemic situation which occurred, right? So it results in major epidemics as well as pandemics. So are these pandemics very frequent in occurrence? No, these pandemics, when we talk about influenza A, it is less frequent in nature, and they can occur in and around 20 years. So they are less frequent and their occurrence is around more or less near about 20 years, right? So these are all important one-liners resulting into pandemics. Which influenza genera is associated with pandemic? It is Influenza A, right? So we have already discussed about genetic reassortment, which is occurring because of the total swap in segments. So this is virus A, virus B, and some segment is from this B, some is from A, leading to discontinuous variation. There's no pattern in segments, right? There is a different pattern which is followed altogether, leading to formation of a very new different virus, which is influenza virus. And this genetic reassortment not only occurs in influenza virus, another one-liner question, genetic reassortment takes place only in viruses which have segmented RNAs, right? So what are the important segmented RNAs where genetic reassortment can occur? It can occur, firstly, of course, it is influenza virus. Then we have rotavirus and hantavirus. There is swapping of RNA segment. So there is reassortment leading to formation of a new virus. Discussing about the important pandemics which have taken place over the years and decades. So the most important ones that you need to remember from this table are 1918-19, that is H1N1 Spanish flu, also known as Spanish flu, and it was the swine flu strain. And it was the most severe pandemic in the history, which led to around 50 million deaths. And what were these deaths associated with? It was not just the influenza viral infection, but it was also because of secondary bacterial pneumonia. And another important pandemic which gets asked in your exams is this 2009, most recent pandemic which took place. It is the H1N1 PDM09. So PDM stands for pandemic. This is the most recent one and it gets asked in your exams. So now talking about some two important strains, avian flu and the swine flu strains, and we should be discussing little bit about them. So now this A, of course it is influenza A, H5N1, this is the most important avian flu strain which has been endemic throughout the world. This is endemic in the world, which has been responsible for n number of outbreaks and certain epidemics, and it has been endemic in the world for over 20 years now. So when was it originated? It was originated in 1997 and the place of origin was Hong Kong. Since it is an avian flu, so the transmission was no human to human transmission. It was only the close respiratory contact, which was with the birds, with the avian strains, which was associated with the transmission. An important feature was that there was no human to human transmission, and it was more associated with mortality. Than morbidity. And the number of mortality, the percentage of mortality was more than 60%. And what was the virulence associated with? This was a highly virulent strain because of higher mortality. And what is this important feature which led to this higher virulence and more mortality? It was, this is important, 𝗕𝗵1F2 protein. This protein was responsible for higher virulence and higher mortality when we spoke about H5N1 avian flu strain. So this is what you need to know from this particular strain. So what are the clinical features? Of course, first thing would be pneumonia, respiratory infection, pneumonia. This is the most important clinical feature in more than 50% cases. And what were the extrapulmonary features? The involvement of CNS and brain. Pneumonia was the most common clinical feature associated with avian flu strain. So these are another important strains which are associated with avian flu. H7N7, it was isolated from Netherlands. H9N2 was isolated from Hong Kong. And H7N9 was isolated from China, and it was responsible for another important outbreak in 2013, recent outbreak there. Not 2013, it was 2018. Now let's move on to most important influenza H1N1, which was associated with a recent pandemic. It caused recent pandemic. It originated in 2009 from California, and it spread to the whole world, worldwide, including India. In around 2009, and it was on June 11, 2009 that WHO considered, or WHO declared it as a pandemic. So most of us know that H1N1 pandemic 2009 was said that it was swine flu. But swine flu is a misnomer when we talk about H1N1 2009 pandemic strain, because it occurs because of genetic reassortment. So what reassortment took place? Which were the animals or humans associated with genetic reassortment? It was because of the strains: 1 human strain, 2 swine strains, and 1 avian strain. So this pandemic occurred because of genetic reassortment between 4 strains. And where did that mixing occur? Mixing happened in pig, and this is the reason why it was said that it is a swine flu type of pandemic. So now, coming towards the transmission, we must be remembering that this transmission was human to human, human to human, and its virulence was less as compared to the avian flu. And why is that virulence less? Because of the absence of this 𝗕𝗵-F2 protein. Absent. This was absent, so naturally they were less virulent. That strain was less virulent. And it was associated with, of course, pandemic, leading to more morbidity than mortality. So this was all about important pandemics, important strains, genetic reassortment, variations. These antigenic shifts and drifts are one of the most important questions asked from influenza, and what pandemics that took place, and what are the morphological features which had led to such variations in the world throughout, leading to pandemics, right? Thank you.